nostalgia, so I apologize if my air conditioning is loud, because it is on video. But anyway, so to start off DNA sequencing, you have to have PCR happen, which was in the other video, but quick explanation. PCR, you take a strand of DNA and you take a segment from it right here, this sequence that you're wanting to look at, and um, this is like a blown up version of it. So that's what you'd be focusing on for PCR, and PCR amplifies or makes copies of that specific segment that you're wanting to look at. So that falls into DNA sequencing because of DNA sequencing, you're wanting to use multiple um, copies. DNA sequencing can be used for uh, discovering pathogens for an outbreak. It can be used for genetic diseases, discovering them, a lot of different things like that. So, like I said, first step is PCR. PCR has to happen first. The next step is, well, in PCR, you would normally have nucleotides, and these nucleotides are the bases, which are adenine, guanine, thymine, and cytosine. And these are what make up the DNA sequences. CGAT. Yeah, CGAT. So normally you would have these deoxynucleotides and a base. So the deoxynucleotide looks like this. It's upside down. Okay. This is what deoxynucleotides look like normally. So hydrogen, oxygen, carbon, all that good stuff. So this is what it normally looks like. But for DNA sequencing, you have to you would add in what's called dideoxynucleotides, which look like this. So you're probably thinking they look the same. Well, you know, but if someone looking at this, they think those look the same. They pretty much are, except this one has oxygen right here, and this one doesn't. And that makes a huge difference when it comes to DNA, apparently. So, um, when you have normally the deoxynucleotides, which allow for DNA to keep elongating. It's fine, it's married. But then once you add in the dideoxynucleotides, it stops the DNA from elongating any further. So as soon as that oxygen is, or as soon as that is added in, and basically that oxygen is removed, that's when it stops the DNA from elongating. So in DNA sequencing, you would be looking at in DNA sequencing. That's kind of important because that's what determines the different lengths for DNA, which is later used with you take your data later for gel electrophoresis, and that basically looks at the data you have and it makes a DNA sequence out of it. So, kind of jumping ahead of myself. So you have your four bases: cytosine, adenine, thymine, guanine, and when you use the gel electrophoresis, basically what it shows is you would have something that looks like that right there. So, you'd have the blue represents a nucleotide, and then the orange represents when a dideoxynucleotide is added in. So, for instance, in this case, you have one nucleotide, two nucleotides, three nucleotides. It's growing, all is great, and then all of a sudden an adenine comes in, a dideoxynucleotide, and it stops the DNA from elongating any further. So, in that instance, the DNA sequence is only four nucleotides long. This one over here, you can see, it is blue, 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 aka nucleotide, 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 going, 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 and all of a sudden a pink fluorescent color comes in, which means that cytosine came in and stopped it, dideoxynucleotide. So that could mean that, there could be two nucleotides long, it could be three, it varies, that's why they're all different lengths. So when you enter this into gel electrophoresis, it takes it takes what you put in, and every time there's a um, fluorescent dideoxynucleotide in there, it copies that down, like what that is. So it could be, cytosine, guanine, adenine, or thymine. It could be any of those. And so it takes all those and it makes its own DNA sequence. And that's pretty much how DNA sequencing works. Um, so yeah, that's my video. Thanks for stopping.